So now you are going to see about structure of proteins.
Structure of proteins, okay.
So generally in structure of proteins, chemists, what the chemistry people, who chemists will do, right? They will just draw the structure and they will show the protein.
Yeah.
They will be drawing the structure of protein, okay, if it is cyclic or non-cyclic or anything, or they will be showing it, okay,
Yeah.
by a diagram or anything. Whereas physicists who study physics, who do research or generally physics, they view these structures all in 3D dimensions.
Okay.
But here what biologists do for proteins, they are grouped these proteins into 4 levels. That means they are showing it in 4 structures.
4 dimensions.
4 structures, not 4 dimensions. So 4 structures, okay. So primary structure, secondary structure, tertiary structure, and quaternary structure.
Okay.
So here, what is primary structure of proteins? So primary structure of protein is nothing but sequence of amino acids together linked by peptide bonds.
Yeah.
So here, what is primary structure is, so what is the first amino acid, second amino acid. So knowing the sequence of amino acids in the protein, what is the first amino acid, what is the second amino acid, what is the third, and knowing the sequence after this what amino acid will come, that is the primary structure, okay.
Okay.
So that is the primary structure, sequence of amino acids, that is positional information of protein, what is the position of that amino acid is known as primary structure. So here protein is imagined as a line or as a thread, because we are imagining only the particular amino acids.
Okay.
So here the left end, that is the first amino, the left end is known as the first amino acid, and the right hand is the last amino acid. So first amino acid is also known as N-terminal amino acid, and at the right end last amino acid is also known as C-terminal amino acid. So here, if these lines, we are imagining the amino acids as lines, right? The protein as a line. So when these lines are folded in the form of helix, in a right-handed fashion helix, so this, other than, there are forms like threaded and right-handed folded proteins that all are available. So when these proteins are folded, they are known as secondary structure. When these proteins are folded. So the first primary structure is knowing the amino acids, it is imagined as a line, and secondary structure, those amino acids are coiled.
Okay.
So that is the secondary structure. So when the protein chain is folded upon itself, it is known as tertiary structure. Can you understand?
Yeah.
In secondary structure, it is folded in a coil in a right-hand fashion. So if more than one or many right-hand fashions or many proteins combined together, and these folded upon itself, this helical structure, both folded upon itself, it is known as tertiary structure.
Okay.
So it is like a woolen ball, a spherical structure, like 3D coiled and this thing and everything. So this gives a 3D view. Many biological activity of protein takes place in this 3D view only. Okay. So next, the manner in which the spherical structures are arranged in cube or in plate. So actually, can you understand what I am saying?
Yeah.
See here. So in primary structure, we know it is, if this is a protein line, we know it is an amino acid, this amino acid, this amino acid, okay. And in secondary structure, in secondary structure you will be knowing this line is folded like this, coiled. And in tertiary structure, many coiled forms like this are upon together, and it forms a spherical ball-like or wooden ball-like structure. So in here, in quaternary structure, now what happens? Many balls of this, whether they are arranged in a cube-like fashion, each one is a single ball.
Okay, we are zooming in and zooming out.
Yeah, cube-like or plate-like. So it is known as tertiary structure, quaternary structure actually.
So first they are zooming inside. So that is
Yeah.
the zoom in. This is a bit out. This is a little bit out. This is more than that. We are
Yeah.
seeing individuals, everything. Okay.
So this is known as quaternary structure and it is also known as architecture of protein.
Okay.
So if you take an example of human hemoglobin, so human hemoglobin consists of 4 subunits.
4 subunits. Okay.
Okay. So in these 4 subunits, 2 of these are identical to each other. 2 subunits are identical to each other. Hence, 2 subunits of hemoglobin are called as alpha subunits and 2 subunits are called as beta type subunits. Okay. So which together form the human hemoglobin. Okay. So this is the structure of proteins. Primary. Are you clear about primary, secondary, tertiary, and quaternary structures? So next we are going to our next topic, which is nature of bond linking monomers in a polymer.
Okay.
Okay. So how the monomers are linked to a polymer, we are going to study about that. Okay. Discuss about the 8th topic, which is nature of bond linking monomers in a polymer.
Okay.
Okay. So we have already seen that in protein, the amino acids are linked through peptide bond, and in polysaccharides the sugars are linked through glycosidic linkage. So here, what are the how this bond is formed? So what is the nature of the bond? How our monomers are formed into a polymer?
Okay.
So we are going to see about these
Different types of bonds.
different types of bonds. So first one we are going to see about peptide bond, which is peptide bond, which is found in proteins. Okay, between one amino acid and other amino acid. So amino acid, we generally know that the carbon atom at the center and hydrogen atom, alkyl group R, and an amino group NH2 and a carboxyl group COOH, right? So what this bond does? So this bond is between the carboxyl group of the previous amino. It connects between the carboxyl group of the previous amino acid and the amino group of the succeeding next amino acid. So it connects these two, carboxyl group of previous and amino group of next one. So how it connects these two? By release of a water molecule, which is known as dehydration. So after release of water molecule, here H2O is water, right?
Yeah.
So hydrogen and oxygen from this and the hydrogen from here will go out as water molecule. So the remaining will be CONH bond.
Okay.
Okay, so this bond, this is known as peptide bond, or which is known as
Which is from us?
which are present in these proteins. Okay. So next we are going to see about the glycosidic bond, which are found in polysaccharides between one sugar and another sugar. Okay. So here also it is a dehydration process, as here it is dehydration. Okay. So here also this bond is formed by dehydration, release of a water molecule. It occurs between 2 carbon atoms. That's all. Okay. So this is the glycosidic bond which occurs between sugars. So next we are going to see about nucleic acid. The bond forms, how the bond forms in nucleic acid. Okay. So in nucleic acid, which may be DNA or RNA, the bond formed here is phosphodiester bond.
Okay.
Okay. What is phosphodiester bond? It is because here this phosphate group attached to the 2 sugar groups through ester bond.
Okay.
What this phosphate bond do? It attaches to the 3rd prime carbon of the previous sugar moiety and the 5th prime carbon of the succeeding sugar moiety. 3 prime, the 3rd carbon and the 5th prime carbon. Okay, so it connects these both and the phosphate bond is present.
Okay.
So this is known as ester bond.
Okay.
Okay, so ester bond is nothing but phosphate and sugar moieties. So this phosphorus and sugar moiety together forms the backbone of the nucleic acid.
So it's like a backbone.
Yeah. Okay. So since here 2 ester bonds are formed across a phosphorus, it is known as phosphodiester bond in nucleic acids. Can you understand? Okay. So we have seen secondary structure of proteins, tertiary structure of proteins and everything. Here in nucleic acid, which may be DNA or which can be RNA, here also there is a secondary structure which was proposed by Watson and Crick.
Okay.
So therefore it was known as Watson-Crick model, or it was also known as double helix model, double helix structure. So in DNA it is double helix structure, which is anti-parallel. Anti-parallel is nothing but they are perpendicular. The 2 strands are perpendicular to each other. Like if one strand is this one and the other strand will be like this. So they are perpendicular to each other.
Like DNA.
Okay. Yeah, DNA is a double helix structure.
But it is spiral.
Yeah, it will be spirally. It will be.
Okay.
So here in the DNA are antiparallel. They are perpendicular to each other. Each turn is perpendicular. They are coiled in a right-handed fashion. Okay. So nitrogen bases. So we have seen in nucleic acid there are three parts: phosphorus, sugar, and nitrogen bases. So where does these nitrogen bases are? They are perpendicular to the backbone. They are like this.
Yeah, like DNA.
Yeah.
We have a round thing in those.
Yeah.
Okay.
So in each one there will be 10 base pairs.
Okay.
Okay. So what are base pairs? Base pairs are of 2 base pairs. If 10 base pairs, there are 20 nitrogen bases. Okay. So it is 10 pairs. So 10 pairs which includes 10 lines like this. So one line, a bond is formed between, one line is composed of 2 nitrogen bases. One line is composed of, one bond is composed of 2 nitrogen bases. Okay. So bond between, what are nitrogen bases? We have already seen that adenosine, guanine, cytosine and thymine. So here with adenosine, thymine makes a bond.
Okay. With
a double bond, 2 hydrogen bonds. And with guanosine, cytosine makes a bond with this, which is a triple bond, which is hydrogen, triple hydrogen bond. So always this adenosine bonds with thymine, always this guanosine bonds with cytosine. Okay, purines with pyrimidines, okay. So like this only bond is arranged. So if you see in this one strand is that A double bond P, G triple bond C. It can have any order, okay, based upon the DNA. So it is like this. So one line is this one, other line is this one. Can you understand?
Yeah, it goes like that. It goes on. The next line it will be AAC, AT, and the next line it will go GC.
No, they do not have a repeater. They do not have a perfect structure. They can be AT, GC. They can be
it's a random.
They can be random.
So with that only they find this thing.
Which thing?
DNA. With one person and another person.
Because of these there are differences. Okay. So here each step if you move on from AT, if you jump from one line to the other line, each step is 36 degree. It turns 36 degree each step. Okay, it turns like this 36 degree. Okay. So one full turn of to bring
like a spiral.
One full turn needs 10 steps. That means 10 base pairs.
In the one full turn there will be 10 things. 10 GC.
Therefore 10 base pairs. So one step it is 1 base pair, and 10 steps it is 10 base pairs. Okay. So here it is of length 3.4 angstrom.
Okay.
Which one? One step is 3.4 angstrom. Rise per base pair. For one step it is 3.4 angstrom. Angstrom is nothing but 10 to the power minus 10 meters.
So rise by base pair means the space between those things.
Yeah, the length.
Yeah, that's what I say. The space length.
Yeah.
Okay.
Length. So this length is 3.4 angstrom. Angstrom is nothing but 10 to the power minus 10 meter. Okay. So pitch is nothing but one full turn, which comprises of 10 base pairs, so 10 steps. So this pitch is 34 angstrom. Into 10 if you multiply, it is 34 angstrom. Okay. So this length is 34 angstrom. So the
per pair it's 3.4 angstrom.
Yeah.
Okay.
So this kind of DNA is known as B-DNA.
Okay. Okay.
So this is about the nucleic acid and the bond formation inside the nucleic acid and inside the secondary structure of the nucleic acid, which can be RNA or DNA. So if you see in case of this is only in case of DNA. In case of RNA, thymine will not be there. Instead of thymine, there will be uracil. Yeah, this is RNA structure. If it is DNA, the adenosine will bond to thymine. Okay, can you understand? If it is RNA, uracil. If it is DNA, it is thymine. So then we have seen various bonds which can be peptide, glycosidic and nucleic acid bonds. Okay, so this is the nature of bond linking monomers in a polymer. So next we'll see next we'll go to the next topic, which is dynamic state of body constituents. Okay, which is dynamic state of body constituents, concept of metabolism. Okay. So here biomolecules, various biomolecules, they are constantly changed to other biomolecules.
Okay.
In our body. Many molecules are changed to other molecules, other biomolecules. Many are changed, like for example, so this is known as metabolism actually. So it is constantly changing. So for example, from amino acid, release of CO2 gives rise to amine.
Okay.
And for example, in glycosidic bond between sugars, if you hydrolyze glycosidic bond, it gives a disaccharide.
Okay.
So again, the disaccharide produces glycosidic bond. So like that, it forms a continuous mechanism like a traffic jam. Everything this goes this reaction will goes that way, and the same reaction will go to some other way. So many diversities and many biomolecules change to other biomolecules, and that biomolecules come back to its own position. So many metabolisms occur in our own body itself.
Okay.
So this metabolic flow is known as dynamic state of body constituents. Okay, state. So here dynamic state is nothing but a state of balanced position. So if you excess of nutrients are stored as fat, whenever the body needs the fat are utilized
for energy.
Yeah. So this is also an example of a dynamic process.
Okay, it's converted to energy.
Yes.
Okay.
So these are going, even though traffic jam, it is going in a smooth manner. Okay. So every chemical reaction is catalyzed reaction. Catalyzed reaction is nothing but a reaction takes, catalyzed means it uses some catalyst. By these catalysts, it performs their reactions very faster. The rate of reaction is very less.
Fraction of second.
Yes, within fraction of seconds the reaction is done.
With
the help of catalysts.
They'll convert.
Yes.
Themselves. Okay.
So some catalysts are made up of proteins which are known as enzymes. So these also catalyze some reactions. So this is the dynamic state of body constituents, the concept of metabolism.
Okay.
So this is the topic.
That's it.
Okay. So next we'll go to the 10th topic, which is metabolic basis for living.
Okay.